All right. I think we'll go ahead and start. I'd like to welcome everyone to our second Voices of the River webinar, hosted by myself, Jennifer Lanham, and Sam Frith of the Evenlode Catchment Partnership. Our aim is to tell the story of the Evenlode and to highlight the actions of the many people behind the recovery of our rivers. This webinar is being recorded, so please do put your computers on mute, and we will have a questions session at the end. So please do feel free to pop them into the chat if you don't want to lose track of them, and then we'll address them at the end. Today's presenter is Elista Penkova. She is an evolutionary microbiologist, now transitioning to become an environmental epidemiologist. She's researching bacterial resistance to antibiotics, and her goal is to produce research that offers compelling evidence for policymakers, ultimately aimed at safeguarding public health. Her research is titled Poo Sticks, and she's here today to tell us all about it. Welcome, Elista.
Thank you. I just probably need to correct you. I'm not a PhD candidate yet. I'm just a student at the moment. A candidate is when you're finished. I'm nearly not as finished as I should be, or as I'd like to be. Can everyone hear me okay? I've sat in the seminar room to use the recording, to use the audio, but I'm not sure if you can hear me well enough.
Yeah, I can hear you fine.
Cool. So I'll share my screen. Oh. Okay. Can you see my screen?
Yeah.
Yeah.
Yep. Okay. All right. So, hello everyone. My name is Elitsa. I'm a PhD student at the University of Exeter. I'm based at the European Center for European Center for Environment and Human Health, which is based at the University of Exeter Medical School, and I study antibiotic resistance in UK rivers. But instead of introducing antimicrobial resistance first, I would like to tell you why antibiotics are really, really important. And I'll tell you that by sharing a bit of a personal story. So this is a photo of my mom and I when I was 5 months old. And this is actually the only photo that we have from that time, because just after the photo was taken, my mom got really, really ill. She got ill because of something that I see quite often today with now that most of my friends are mothers. They don't really like to ask for help. So taking care of my then toddler sister and myself, my mom got basically really, really exhausted. Her immune system crashed. And as a result, bacteria, which had been hiding in her body for years, finally saw an opportunity. So at the age of 27, my mom developed tuberculosis. And this is not a disease that we often talk about today because of successful vaccine intervention strategies. But it has been estimated that even today, a quarter of the world's population still carries latent or dormant TB bacteria, just like my mom did. And only about 11% of these ever develop the disease. So you can argue that my mom was really unlucky to develop this illness, but I would actually argue that she was really lucky because her infection was susceptible to treatment, which means that for 6 months she was able to take rifampicin, which is a particularly nasty type of really potent antibiotic, but she was able to get better. And I got to grow up with the mom, and now my niece gets to grow up with her grandma. Other people are not so lucky. So each year it has been estimated that about 450,000 cases of new reported tuberculosis cases resistant to antibiotics. So it's a really huge issue, but this is not a tuberculosis issue, and I'm not here to talk about tuberculosis. This is a global health public crisis issue. But more and more bacterial infections are becoming resistant to antibiotics, and as a result, people are increasingly losing the battle against infectious disease. And it has been estimated that if this issue is not tackled or it's not somewhat controlled, in about 25 years or so, we're going to see bacterial infections becoming once again the leading cause of death worldwide. So why are bacteria evolving resistant? So why are they becoming resistant to antibiotics? Well, To address this, I would like to let you know what antibiotics are. So these are molecules produced by microorganisms, including by bacteria, and they use these molecules to kind of communicate with each other or to fight each other off for space or resources. And what we humans did was simply identify which antibiotic molecules are effective against the specific bacteria that we were interested in. So any bacteria capable of causing disease in us or the animals we care for. And once we did that, we were able to figure out how to weaponize these and mass produce them to treat bacterial infections, which in turn obviously revolutionized modern medicine by allowing doctors to safely perform routine and life-saving procedures such as hip replacements, such as cesarean, organ transplant, or chemotherapy. But we need to understand that bacteria have had to live with antibiotics long before humans ever existed. Obviously not in such vast quantities, but nevertheless, they have had billions of years to evolve coping mechanisms or resistant mechanisms against these antibiotic molecules. So how does resistance emerge? Well, one of the simplest ways in which the evolution of resistance happens is by maintaining large and diverse populations. So if you think about any population, so be it bacterial, human, dog, cow, whatever, within any population, there are slight differences between each individual within that population, right? So for example, this was very obvious during the COVID-19 pandemic when we had to deal with a virus that none of us had ever encountered before. And some people got severely ill. Some people only had mild symptoms. Some people sadly lost their lives and others didn't experience any symptoms whatsoever. So this was despite never having to deal with this particular virus before. So in a similar way, if we imagine a bacterial population, we can expect to see subtle differences between each individual cells. And these differences don't necessarily make a difference in an environment where all things are equal. And we can expect these different individuals to pop in a population at random, and in the same way, of course, become extinct at random. But if we suddenly drown this population in a toxic substance, such as an antibiotic, then those cells whose differences mean the antibiotic is not able to recognize them anymore and binds to them, then this means that these few different individuals are now able to survive and grow as well as replicate despite the presence of the antibiotic. And so every time we take antibiotics, we are essentially increasing the natural proportion of resistant bacteria simply by wiping out all the rest of the bacteria within the population. So there are 2 points I'm trying to make here. First, resistance occurs naturally and spontaneously. And the second one is that for the evolution of resistance to occur, which is a change not in the individual, but a change in the population as a whole, we need to have selection. And the primary selective driver for the evolution of antibiotic resistance is the use of antibiotics. Although there are other drivers, but I won't get into those today. So the process of maintaining natural variation and relying on chance to be a little bit different enough for the antibiotic not to be able to kill you anymore is kind of the simplest form of resistance evolution, but it's not the only one. So bacteria are actually able to change genes, so they can literally extend these kind of appendage-like pili where they can share genetic information between a donor cell and a recipient cell, essentially transforming these bacteria from a susceptible to a resistant bacteria, which means that essentially once a resistant mechanism emerges, it can really easily spread across the population. And a bigger issue with this is that bacteria can also accumulate various resistance mechanisms rather than actually relying on chance. They can accumulate them at the same time. And this is basically how we go from a bug to a superbug, which is a bacteria resistant to either multiple types of antibiotics or to all antibiotics that we currently have in use. And on this note, I would like to move on to the topic of my PhD, which is looking at antimicrobial resistance in rivers in the UK and whether that poses a risk to the public. So We know that it's really important for swimmers to be able to go for a swim in a nearby river or a lake across the country, as people do report that this helps them deal with things like grief, like depression, or coming to terms with an illness diagnosis. And of course, for most people, it's simply quite joyful to immerse themselves into these natural environments and being kind of able to reconnect with nature and de-stress, which is also really important. Of course, the concern here is that rivers and lakes and other freshwater environments, as I'm sure you're all aware of, are contaminated with sewage pollution and animal runoff, among other things. And with these sources of pollution, the problem is that we're introducing fecal pathogens, including resistant pathogens, which are becoming more prevalent. In addition to bacteria, in addition to bacterial sources, along with this waste, we're also introducing the selective drivers themselves. So we're also introducing antibiotics. And every time we do take antibiotics, there are about 70% of these are become excreted through our urine and feces because we're not able to metabolize these. So we are basically not discarding them, that's not the right word. We're excreting them, that's the word. We're excreting antibiotics when we take antibiotics through our urine and fecal waste, and that eventually ends up in the sewer system, and it can either end up in the rivers in a raw state, or it can be treated to a certain level. So this is kind of why this is a concern, but what is the evidence that this is something that we need to worry about? Well, there is fairly strong evidence that the levels of antimicrobial resistant bacteria and genes are in fact linked to pollution, and we see higher levels of resistance downstream of something like an effluent discharge point or a farm relative to upstream of that source. In addition to this, we also know that freshwater swimming has previously been identified as an independent risk factor for developing resistant urinary tract infection, with a study from Norway, which reported that patients carrying resistant bacteria swam twice as frequently as patients who carried susceptible bacteria. In addition to this, we also know that freshwater swimming has been estimated to account for about 6% of the cases of antimicrobial resistance acquisition by the general public over the summer months. And I actually haven't included this study here, but there was a really interesting case of sepsis after a near-drowning event in a French river, where luckily the patient was survived, but the causative agent was identified as a resistant Enterobacter bacteria, and identical isolates were actually found in the river where the patient was taken out from. But of course, this is just a one-person case study. But we don't actually know what the risks of exposure to resistant bacteria from swimming in rivers are. But we know from previous work on coastal waters that surfers were found to be 3 times more likely to carry resistant bacteria in their guts compared to people who don't go into the water. In a more recent study, found actually that in coastal swimmers specifically, along the coast, the Irish coast, they were found to be less likely to carry resistant bacteria in their guts. And it's very likely that this difference is due to the nature of the two sports. So if you think about it, surfers are more likely to fall accidentally. On average, they ingest unexpectedly about 170 ml of water. And swimmers, on the other hand, have a lot more control during swimming. So on average, it has been estimated from swimming in pools, not in the coast, that swimmers ingest about 16 ml of water during a 45-minute swimming session. So a higher volume of water means that there's more opportunities for encountering these resistant bacteria, which means there's more opportunities for colonization of the gut. But if you think about it in contrast to coastal swimmers, river swimmers are actually exposed to much higher levels of pollution, where the potential in these environments for dilution is it's significantly reduced. And this often leaves freshwater swimmers and bathers frequently exposed to unsafe levels of pollution and other associated pathogens. So to address this, over the course of the summer, I'm recruiting volunteers to take part in 2 studies. So the first one is a poo stick study. Here I'm asking both river swimmers and non-swimmers to send poo samples for analysis back in our laboratory at Penryn. So this happens over the post. So volunteers from all over the country are able to take part here. And the purpose of the poo stick study is to understand how river swimmers acquire antibiotic-resistant bacteria in their guts. So each sample would first be screened for the presence of cefotaxime-resistant E. coli. And E. coli is a bacteria which is normally found in our guts. It's part of a healthy microbiome and it's involved in digestion. But E. coli is also what we call an opportunistic bacteria. So while it can remain in the gut of healthy individuals, typically in a harmless state, it can become pathogenic if it has the opportunity to do so. And cefotaxime-resistant E. coli is a particularly nasty type of resistant bacteria, which is listed as in the critical group of the priority pathogens by the World Health Organization. And they kind of describe it as bacteria which poses the highest threat to public health due to limited treatment options, high disease burden in terms of both morbidity and mortality, and increasing trends of antimicrobial resistance with few or promising candidates in the pipeline. And infections with pathogens in these groups are also uniquely difficult to prevent and highly transmissible. So because E. coli is part of our normal gut microbiota, we can actually carry this bacteria asymptomatically. And the concern here is that we can spread this to individuals who are more vulnerable to infection, and we're putting them essentially at risk for developing an infection with already limited treatment options. So in addition to screening for cefotaxime-resistant E. coli, I also want to look at the bacterial community in the guts of swimmers and non-swimmers. So I want to look for patterns, for example, between individuals who are colonized or not colonized by these bacteria, understand whether swimming in rivers has an effect on the risk of carrying resistant bacteria and genes, or is there something else going on, for example. This is kind of the part that I'm most interested in because we don't really know much about the gut microbiome, so it would be really interesting to see what comes out of this study. So any individual who is healthy, who swims in rivers, and those who do not swim in rivers across the UK might be eligible to take part, although here it's important to know that there are some additional criteria, such as, for example, you shouldn't have left the country. No, actually, you shouldn't have left Europe within the last 6 months. You shouldn't be on antibiotics or living with someone who is on antibiotics. You shouldn't be on any medication, any sort of chronic condition and stuff like that. I can explain why later if you have questions about that. So yeah, we're looking for not just for swimmers, but also for non-swimmers. And we actually almost filled out all of the spaces for swimmers, and we are desperately needing more non-swimmers. So if you do know any people who do not like to swim, please let them know because we need their poo samples. Sign up. You can find the study by Googling the Poo Stick study at the University of Exeter Medical School, where you will find a link, which is right here. So when you click on this link, you'll have essentially a questionnaire, which will tell you, ask you all of these questions to determine your eligibility. So once you have determined whether you're eligible or not, you'll be able to provide your email, your name and address. Please provide both your full address, including your postcode, because I can't guess them. I don't know if you can. If someone can let me know, please do. But I've had a few people who have just sent me the first line of address, and I don't know where to send that kit to. Anyway, so if I get the address and name, I'm able to then send you a kit wherever you are in the UK. So the kit includes everything you need to know to take part, including a prepaid return box. So there's no expenses for participants. So for river dart swimmers, specifically, there is an additional option to request. This is actually no longer the case. I've taken that out of the questionnaire because we've filled out all of the spaces for river dart swimmers, but I'm just going to let you know what this is about. So with river dart swimmers, they were able to request 6 kits rather than 1, and they're able to send these once a week or essentially 6 times throughout the summer. And throughout the summer, I'm also collecting samples from the water, from the River Dart, and I'm isolating resistant bacteria from that river. And then by comparing isolates from the river to those from the fecal samples, I might be able to establish links between water exposure and colonization of resistant bacteria. I'm going to stop here with the Poo Stick study. The second study that I'm recruiting for that is not really talked about a lot is the National Health Snapshot. So because the Poo Stick study looks at bacteria in healthy individuals, as I said before, these bacteria we're looking at can simply be a part of a normal gut microbiome, which means that people can carry these resistant bugs in their guts without necessarily having to experience the consequences of it. Of course, provided their immune system is working fine. So colonization status will not tell us a lot about the risk of experiencing poor health outcomes after exposure to a freshwater environment. And this is something that we sometimes hear about on the news, which suggests that exposure to polluted waters can have significant effects on the health of the growing community of freshwater swimmers. But we don't know how significant these risks are, because the symptoms swimmers experience are typically mild and they're often underreported. So because, this sometimes, I don't know, at least I do that. If I have like a headache or something, I won't go to the GP to report that. I'll just wait for it to go away on my own. So the idea of this is to kind of make an assessment and understand what symptoms river swimmers might be experiencing and how different these experiences are from people who don't go into the water. So this study is it's an online questionnaire, so it takes about 10 minutes to take part. It's open for participants now, along with the Poo Stick study, and you're actually able to agree for your data to be linked from this survey to the Poo Sticks results. And the consent for that is as an additional option in the consent form for the Poo Stick study. You don't have to worry about that if you're just taking part in the National Health Snapshot. So it's a 10-minute questionnaire, roughly, depends on whether you're a swimmer or not, or dyslexic or not as well. Anyone over the age of 16 living in the UK is able to take part. And again, we're hoping to kind of understand what illnesses are freshwater users at risk of experiencing and whether these differ the longer people stay into the water, for example, or under different weather conditions. And I should mention that I've written here bathers and non-bathers, because as opposed to swimmers and non-swimmers, because if you're a kayaker or I don't know, if you're participating in any other type of water sport, you're still able to take part in here as a bather. I do get a lot of questions from kayakers specifically. So if you're a kayaker, this can be the study for you. And this is pretty much it. This is my supervisory team. And there's a lot of people who have helped do this. It's definitely not been a one-man job. So yeah, thank you very much. And if you have any questions, I'm happy to take them now. No, actually, stop.
Thank you so much for that. That was brilliant. I was going to mention that actually I am participating in the survey as a swimmer, and I'm really, really excited about that. Can you tell us how many people you're looking for to participate?
Oh, sorry. Sorry, I think I accidentally. Hold on, let me just fix my camera. Sure. Full share, stop share. Okay. There we go.
I was going to show everybody my little test tube samples that I've received actually, which is really, really great. I was excited to get my parcel in the post. But so how many people are you looking to participate? Are you looking for equal numbers of swimmers and non-swimmers?
Yeah, so I need a minimum of 150 people to take part as swimmers and 150 people as non-swimmers. And so far, about 650 people have tried to take part. From these, 280 were successful. 200 of those were swimmers, and about just over 50% were non-swimmers. But the thing is that I think because we got a lot of media attention, a lot of people signed up without realizing that they were not eligible for it. So a lot of sea swimmers ended up signing up, and they're emailing me, asking me to return the kit and stuff like that.
Yeah.
Which is totally fine. I'm happy to get them back. But it's just the whole, I just feel bad. We should have made it really clear. But the thing is that because we're recruiting alongside another study for sea swimmers, and the media really kind of said river and sea swimmers together. So that was something that we should have foreseen. But yeah, for the questionnaire, we're recruiting about 450 people per group, so 450 swimmers and 450 non-swimmers. And so far, I've only had about 300 attempts to take part in the study. From these, I haven't even looked, because it's an online questionnaire, so they can just accumulate. I haven't actually looked how many of these are swimmers, how many of these are non-swimmers, and how many of these have actually finished the questionnaire, because a lot of people check it out, have a look, try it. It might be a bit longer than they expected, and they can quit doing it, which is fair enough. I've done it a lot of times, so I understand.
And are you just looking at E. coli, or are you looking at, like, I've heard that plastics can have an impact as well on your immune system as far as carrying bacteria around your blood and whatnot. So are you looking at plastics or anything other than your?
So I'm just looking at bacteria. So I'm looking at E. coli and the bacterial community within the fecal sample. We have another person in the group that looks at microplastics as vectors for antimicrobial resistant bacteria. But it's really difficult to, like, we can make it a really, really big project and do it really poorly, or we can break it down into smaller bits and do it fairly confident in our methods, which is why each of us focuses. In my group, all of us work on antimicrobial resistance, and each of us works on a very tiny component of the same issue. So it would be really helpful if I could have all of them here in the webinar and we can talk about our research. That would be really helpful. But yeah, I do work only on bacteria.
Excellent. Does anybody else have a question?
I was going to ask, do you foresee an issue with the samples you get if people are taking probiotics?
No, I don't see why that would be an issue. So the thing with the gut microbiome, it's very, very dynamic. So if what we're doing is a cross-sectional study, which means that we're taking a snapshot of what is going on right now. So if someone tests positive now, we can sample them tomorrow or in a month from now, and they might not have the same bacteria. So I kind of knew this in theory, but with the River Dart participants, that became very, very obvious. I don't know if anyone else is like this, but when I don't quite understand something unless I've seen it. So when I started receiving samples from River Dart participants once a week or once every couple of weeks, then it kind of became really obvious. I haven't seen any cefotaxime-resistant bacteria in them, but there are other bacteria which are resistant to antibiotics, which is normal. There can be part of our gut microbiota that can carry intrinsically, so they naturally carry resistant mechanisms against these type of antibiotics. So we see certain types of bacteria pop up on the plates, and then in a couple of weeks, they're gone. That's how dynamic it is. So when I say a cross-sectional study, I mean that we're just taking one sample at one single time point. That's why we need so many people, so that we can have an idea of how confident we can be in these data. But if we take the exact same data, if we take samples from the exact same people a month later or a couple of months later, we might see something totally different. Sorry, was that your question? I think I rambled too much.
No, that's fine. I was just talking about, is there an effect of probiotics in terms?
Oh, yes. Probiotics. I've included this as a question. Actually, a lot of people volunteered their whether they're taking, so I had to change the questionnaire essentially. With the Poo Stick study, you get a questionnaire into tubes, which you can add your sample, and then the questionnaire asks you about where you've been to, what types of foods you've eaten, where you've traveled, etc. And initially, I wanted to include only supplements. I wanted to see whether people were taking supplements because these are typically quite high numbers of bacteria in them seeded in your gut. So the purpose of them is to colonize your gut. So I wanted to see whether if we can control for this, for taking supplements, probiotics as supplements, whether this would have some sort of impact on whether people are colonized or not. But people started volunteering their information by adding in brackets, whether they've taken yogurt or kefir or sauerkraut and stuff like that. So now I've added this as an option in the questionnaire. So people who receive kits now will actually have options for whether they've eaten probiotics as food or prebiotics as food, or whether they've taken them as a supplement, just to make it a bit clearer. Because I think when I eat a lot of live foods like kefir and sauerkraut, and I do believe that affects your gut more than water exposure, for example, because we're taking such vast quantities of these, and they're going straight into the gut. And a lot of these do survive stomach acid. It's not, we do think that stomach acid kind of dissolves everything, but it doesn't. What we definitely affects our gut. And what we drink isn't.
We've got a question from Janine saying, when do you think you're going to be able to report your results?
As soon as I get enough non-swimmers, essentially. Because I've sent out about 200 kits to swimmers. And if I get these results, if they return the kits back until the end of the summer, hopefully, I will have data on swimmers, but I will have nothing to compare it to. Okay. So far, I have the same amount of swimmers and non-swimmers return the kits back. So far, it's kind of 50-50. I only have, granted, it's only 25 people per group, but I see there were 2 people who tested positive from each group.
Okay. Wow.
That's really
so you're the first ones to hear the first results.
Yeah, that'll be good. We'll push it for you. I know plenty of non-swimmers, but I know lots of swimmers as well. So yeah, start
really
asking
helpful,
our friends and family that like to stay on dry land.
And Elista, why did you decide on the River Dart for your additional kind of study, your more concentrated study?
So we started talking to Hannah from the River Dart. She's a River Dart lead. Probably 2 weeks into when I started the PhD. So she has been very, very kind of not vocal. What's the word? She's been advocating for the River Dart from the year before my PhD started, and I don't know if she contacted us or my supervisors contacted her, but we've been talking since the very beginning of the PhD, which was 2 years ago, and we had this idea. Originally, the study was only going to be on the River Dart people, on River Dart swimmers, because there was such a huge interest from River Dart swimmers, and because the River Dart is 2 hours away from me, I'm actually able to go and sample. I can't sample the whole of the UK. That'd be great, but I can't do it physically. I'm struggling with the River Dart, to be honest. It's a lot to constantly be out and about. So yeah, it's fairly close. There's a huge population of swimmers, and yeah, there were quite a lot of people who were keen to take part. But we decided at the end it would be better to be a nationwide thing because mostly about the eligibility criteria. I think I was really worried that even if there's a lot of people who want to take part, a lot of people wouldn't be eligible. And Totnes, there might be a lot of swimmers, but considering I need 300 people to take part, it's kind of eliminating most of the town. So yeah, that was why we had to expand it, which I'm really happy to that we did this, because especially when we started advertising it first. So I've been advertising it on Facebook because the media refused to
really
publish it. They didn't refuse to. I think they weren't allowed to publish anything that wasn't relevant to the election. We didn't plan on the election, so that was a bit of a curveball. So I started advertising it on Facebook, and because of that, I was getting such an even, lovely spread of swimmers, non-swimmers from all over the country, which was really nice. It wasn't very concentrated because the surfer study I was talking about earlier, that was really concentrated in the southwest, the southwest of England. So while we're talking about the UK surfers, obviously most surfers are down here anyway, but it's just looking at the very scattered, very specific type of population. While with swimmers and non-swimmers across the whole country and having a nice spread across the country, I think it provides a really good snapshot of what the situation is, particularly even in non-swimmers. We don't really know how many people are carrying these bacteria in the general population. I know that the UKHCA is running a study right now, but we don't know how far along they are on how many people in the general population carry these type of bacteria in their guts.
Right, okay. And I was wondering, so with a lot of our research, outreach, we recommend people do not actually go in the rivers anymore to swim, especially the even load. Can you give us any advice that we can share with the public as far as what they might be able to do to kind of improve the situation? I think I read something about, taking your whole course of antibiotics, for example. Is there anything else that you think would really help that people can do? We want to empower people to try and make positive change as well.
So with antibiotics I feel really uncomfortable around antibiotics. Obviously, they're really, really helpful. But we have grown into, it's really tricky. I don't want to say don't take antibiotics if you can avoid it. If you can avoid it, do. Don't go into having a sepsis just to avoid antibiotics either. It's really important to use antibiotics when we need to. A lot of the times we are prescribed antibiotics for diseases that it might take us maybe 3 days longer if we didn't take antibiotics to overcome it. But it's just really tricky because when it comes to your own life, obviously you're always more aware, or especially when it's your child, you would never want to put them at risk. But then again, if you use too many antibiotics, you are putting them at risk. So it's a really difficult kind of line to navigate. The thing with using a whole course of antibiotics, I think someone cited me saying this. I don't remember saying this because I might have done, I don't know, but the literature is not quite certain what is the best way of using antibiotics. So some people say use one big dose once. Some people say use a course of antibiotics several times. This is for doctors to kind of figure out, to be honest. I'll leave it to them, but I don't think they agree fully on what's the best course of action. It might depend on the type of antibiotic, on the type of disease. Yeah, but it's really tricky.
When I first came to the UK, and I got a sniffle, we went straight into the GP and we were given antibiotics. And that really shocked me because my experience in the US was if you felt unwell, you would go in and they would take a swab from your throat and then you targeted antibiotics rather than just, here you go. And I was really, really shocked by that because it was just so easy. And I think since then there's been a fair bit in the media talking about overprescribing.
What year was that?
Oh, I'd hate to guess. It was like 20 years ago.
Well, that if you go now, you would struggle to get antibiotics, I think.
Okay.
They're really, really strict. Both in terms of, I think the UK is much better than the US at the moment, especially when it comes to animals, because most antibiotics are used in animals as either as animal feed.
The rearing of them, isn't it?
Yeah, which is completely ridiculous because it's so unnecessary. But what was I going to say? Yeah, in the UK, it's kind of flattened out. So a lot of farmers came together working with vets and they figured out like, hey, we should stop using these antibiotics because of antibiotic resistance. So it was farmer and vet led in the UK, this movement of keeping away from antibiotics. But at the moment, the levels of resistance in farming, I believe, are quite like leveled out. But it's been the product of a lot of effort by farmers and veterinary people.
Okay.
It's the same with the healthcare system.
Yeah, I think the only other thing that I had read just on this before we go to Anne with her question was that I read somewhere that about 70% of the well, because of our metabolism and the way we process antibiotics, we only absorb a small percentage, and 70% is actually excreted out in our urine and feces. And I was really shocked by that.
So this percentage actually varies a lot. It can be anywhere between 30 and 90%. I think 70% is a conservative estimate. What was I going to say? There was a study from the United States that I can't remember what the study was called or what year it was from. I can have a look, but there was a study that kind of found that people who eat animals raised on antibiotics are carrying resistant bacteria with controlling for all other type of confounders, which was a bit shocking because we often hear that antibiotics are not retained in our meat, in our muscle, or in our bloodstream, I guess. But some of it must be, because there was no other explanation for the levels of resistance with that particular population other than just feeding on animals raised on antibiotics. So he's kind of I don't know, it's not talked enough and it's not researched enough, even though there's a rich kind of literature on antimicrobials, but everything moves really, really slowly. It's mostly epidemiological studies we need a lot of studies to confidently say this is coming from this pathway. It's really difficult to claim cause rather than a link, and disentangling links is very, very, very difficult.
Okay. Anne, did you have a question?
Yes, apologies for being late to join you. I was trying to finish off something else and unable to get back to the screen in time. But I may have missed something of what you said. But when you were talking about the national health-wide survey, as it were, rather than the one you were doing, the surfers, you mentioned kayakers, lots of people who are kayakers. Could you just refresh what the criteria are that you're looking for people for that survey, and is that something that we can also just get a sort of summary of things you must have or things you don't bother with applying to do the survey?
Yeah, so for the National Health Snapshot, the online questionnaire, the eligibility criteria is to be over 16, to be living in the UK, and to not have taken part in any water-based sports outside of the UK within the last 30 days. So if you've just returned from France or Spain, you can wait a month because the survey is going to remain open probably until the end of September, but it's looking like we might just leave it open until the end of the year, just because we're not getting enough traction, we're not getting enough people taking part. And you can take part every couple of weeks because the survey asks you about symptoms you've experienced in the previous 2 weeks. So you can take part as much as you like. We have a way of linking your previous answers, so we can create essentially a longitudinal study, like a long-term study of symptoms.
Should we go to Julian next?
Thank you, Elitsa. It's a hugely important subject that you're studying, and as you sort of hinted, we're waging a war on pathogens that we may not win. And that's really terrifying.
I didn't say that.
Okay, you hinted that this was something where we were running out of antibiotics to resolve these conditions. Don't want to put words in your mouth. I'm a public health engineer. Sanitation is close to my professional expertise. And I think it's really important to remember that vastly the biggest gains in human health over the last 150 years, something like 90%, and this is often forgotten, has come about simply through refrigeration and excluding pathogens from our drinking water. Unfortunately, Basil Jett, who was the leader in this country, there were, of course, many other great engineers elsewhere around the world, engineering fine public health systems in the Victorian era and earlier than we had in the UK, tended to overlook the wastewater side. And that's what you're picking up on now. Sam Frith made a very interesting question about the role of probiotics here. We know that natural ecosystems work with probiotic microbial films to keep everything working well. And to come back to my suggestion that we've been waging war on pathogens, of course, all our human interventions in landscapes, particularly through sewage treatment, come about through antiseptic and disinfecting roles. Disinfection is, of course, one of those selective pressures that encourages antimicrobial resistance. I could go on at length, and I won't. I'll curtail it here. Let's just consider the appalling intensive animal husbandry that is taking all these colossal quantities of antibiotics. I think it's something like 70%.
Globally, but the UK is doing much better.
Yeah. You know, why do they need all these antibiotics? It's not just as a growth promotant. It's to keep the poor bloody animals alive in the most appalling conditions. So the sooner we ban this agriculture, the better. If you look at True Cost Accounting, great book from Routledge, it all operates at a horrendous loss anyway. There's a great play doing the rounds, and I'm sure everyone here is aware of regenerative agriculture. This surely is where the answers lie. And back to my profession, sanitation, yeah, we can engineer diverse treatment processes that resolve antibiotic resistance rather than create it, as our present electromechanical sewage treatment does. Anyway, I'll leave it at that. I'll email you. I did a presentation on this for the BMA, and yeah, I'll send it to you, Elitsa.
I do think you're right. I think a lot of the decisions that we make are very short term. We don't really think about if you think about the wastewater industry as a whole. So it was kind of done. There was a very quick decision. Hey, the wastewater is making us poorly. We need to direct it somewhere. So okay, they directed it towards rivers. That's the cheapest way of exporting our waste. We can't really keep it in our houses because it's spreading cholera and other diseases. So that was a good decision, but it was a very short term. We never thought about, hey, you know, that's our drinking water. That's the water that is fresh water. It's a precious resource. It's the smallest amount of valuable, super valuable resource that we have, because most of the water is salt water. It's not fresh water, and we're essentially dumping our waste into this super valuable resource. So we're not. It's the same with farming. Let's take antibiotics because it helps animals grow quicker and it prevents them from, developing infections and so on and so on. That's obviously driven from the market. The people require buying cheap meat, and we can complain about it all we want, but how many of us are actually buying organic? I certainly can't afford it. Sorry.
It's not cheap meat. I'm sorry. The externalized costs of those cheap £4 chickens mean that they're actually more expensive than organic. The whole market is rigged. I recommend True Cost Accounting by Routledge.
I'll have a look.
Yeah, I'll send you yeah, I've lectured at Exeter. David Butler's a keen supporter of. He says our work is ahead of the curve, so I'll send you some of our material.
Yeah, I mean,
so you know, and let's just come back to those cheap chickens. I'm a Stroudie. I don't know if you know about Stroud, but we're a little hotbed of environmental activists, and we had the late Polly Higgins who came up with this eradicating ecocide movement. And she spelt it out so elegantly and so clearly. And before she died in 2015, and this is not a supernatural statement or theological, we can actually define this through DEFRA's 5 new principles. she said we simply have to work for a higher moral authority. And, you know, that's what a mother applies to the upbringing of her child. It's what a regenerative farmer applies to the management of the land. It's not what these poor intensive chicken and other livestock farmers apply.
Yeah.
Said too
Yeah.
much. So we need a huge cultural change in our society. And actually, it's going to be great because all these alternative methods work much better than the awful practices that are going on now. As I say, I'll email some references.
Yeah, I completely agree. I don't mean to blame, but it just seems like everything we end up talking about, it just seems like it's been very unsustainable. It's been developed very unsustainably and without any kind of thought for the long term. And we're getting to a point where we can't really afford it anymore to keep it going that way. So we do have to rethink how we do things.
Fab, is there anyone else that would like to ask a question while we're all here? No? Fab. Well, thank you everyone who attended, viewed, asked a question, and Elitsa for your fabulous presentation. We'll certainly get sharing and ask our friends and family to take part, and you can get your studies well underway and see your results. I'm very excited to see what they show. And yes, we'll be doing more podcasts, we'll be doing webinars, and we'll be sticking them up online for you to watch later or to share with your family and friends that might have missed it. One last question for me, Elitsa, is I'm going to put you on the spot. Can you tell us what your favourite or inspirational book is that you could maybe recommend for us to read?
Oh my God, does it have to be environmental?
No, not necessarily.
I haven't read in a long time something that wasn't related to my project, but I would recommend, I don't know if you probably all read this, Leo Tolstoy's Resurrection.
Okay, I haven't.
It's
But I'm
really
not a big
good.
reader.
So someone recommended, it might be someone from this group, I can't remember, but someone recommended this book about, actually, let me have a look. It was about wastewater, because Jennifer actually talked about wastewater a lot, and I was a little bit concerned that I don't know enough about wastewater to be able to talk about it. And someone recommended a book. I'm just going to have a look at what the title was because I forget. And that was quite interesting, actually. Something about... Oh my God, I can't find it now. I can post it in the chat afterwards. It had to do with the fact that in California, they actually use wastewater, sewage water, they recycle it and they use it. They're trying to get it, to get breweries to buy recycled sewage water, which has been so purified that it's actually cleaner than water that they receive from rivers and from groundwater, which I thought was quite astonishing because, you know, we're kind of dumping our waste into the river and we're wondering how to clean it up. And in the meantime, there's technologies out there that are just making it available for people to recycle water to a state that is purer for drinking. That has literally gone through us. It's just really, I don't know, I thought it was a really cool book. I would recommend that. Oh,
Well,
Purified. Sorry.
Purified
by Peter Annin. There we go.
There you go. Now we know.
Thank
you again
Thank you so much for having
for your
me.
time. No, it was a pleasure. And yeah, we'll see everyone again at the next one. Keep an eye on our social media for the dates. And yeah, we'll look forward to seeing you then.
Great. Thank you so much. Bye.
Bye.
Bye.